And now we come to a very important part of the meeting as well. We heard this before. This will be the rising stars, the future superstars. And this is about the poster session. So we have more than 20 posters outside. And in order to give you a little bit of taste how exciting they are and have you then during the lunch break also visit the posters, the lottery was selecting 10 flashes. So it's not by purpose, it's just by accident. And each of them will get now 1 minute. And I have a friend up there who will, in principle, sound a gong. And then the minute is over, and then the next person starts. So the first person will be Louise Chambord. She gets the magic box and will start. And then the time is running. There will be no mercy, okay? You are happy to applaud when the minute is over, and then the next one comes just afterwards. So, Louise, go for it.
Thank you. So MS is highly heterogeneous, and guidance on when and how to adapt treatment remains limited. Multiscripts aims to address this gap. It is a pragmatic, randomized platform trial designed to generate evidence on decision-making strategies. The principle is simple: to treat as less as possible, but as much as necessary. So the first cycle evaluates 6-monthly serum NFL monitoring versus usual care to support treatment escalation or de-escalation. Essentially, we want to know if intensive biomarker monitoring can help the patient feel better. So Multiscript is embedded in the SMSC across 8 different centers with 918 patients recruited in less than 15 months. As we can see, we are already well ahead into the study, and if you want to know more about the project, you can come and see my poster number 6. And thank you to all of the participating centers for their commitment.
Wonderful. Maximilian.
Okay, could I go?
Yes.
So
Time is
thank
running.
you very much. So I'm happy to present this cohort with a data set of 259 patients who got a lumbar puncture between 1998 and 2008. And we now retrieved a complete clinical follow-up, including notably longitudinal EDSS assessments over up to 25 years. And we see first that the trajectories are widely heterogeneous, some patients having long-term stability and others experiencing severe EDSS progression over time. We see that baseline brain and spinal cord MRI lesion load is associated with clinical disability and notably time to EDSS 4 and 6. We also see that positive oligoclonal bands at baseline are associated with negative outcomes on the long run. So all in all, this is a unique data set with still available samples on serum and CSF for further research into predicting disease progression, and for more information we can talk later at the poster.
Thank you. Wonderful. Stephanie Martini from Bern.
Thank you. The prevalence of MS is quite heterogeneous globally, and there are a lot of factors that might at play here. Diet is interesting because it's modifiable by individuals, and patients ask about this actually quite frequently in consultations. And there is some evidence for association with certain dietary components, such as dairy, or compositions such as the Mediterranean diet, but there is still a large evidence gap. So with the support of the Swiss MS Society, we're now working on a comprehensive exploratory analysis of diet and MS on a global scale using openly available data. And the first result is that a high consumption of vegetables is associated with a lower prevalence of MS at country level. There's more to come. Please do stop by our poster for next steps and discussions. Thank you.
Thank you very much. Wonderful. Alexandra.
This. Hello. So
The green one.
exactly. So we
This is the magic box here.
Yeah.
This one.
We investigated if cognitive impairment is associated with future disability progression in MS. And we included 398 patients from the SMEC with complete neuropsychological test battery, the BICAMS. Cognitive impairment was defined using the Z scores based on regression-based norms developed by Penner, newly published, and the follow-up time was a median of 7 years. we saw that more than half of our people with MS had cognitive impairment in at least one cognitive domain and over 1/3 in multi-domains. And importantly, these patients had a twofold higher risk of developing PIRA over the follow-up. And these results stay stable even after we adjusted for EDSS at baseline, age, sex, fluid biomarkers. All in all, this suggests that testing is important because cognition might be an important
And it's
prognosticator.
over. Thank you.
And Matthew Fox.
Johanna.
Works. Perfect. So we measured a panel of 23 promising pre-selected biomarkers in the CSF and serum of more than 300 MS patients uncontrolled. And what we found was that in the CSF, cytokines CXCL13, CXCL9, interleukin-12b were highly increased and were associated with focal inflammation like relapse activity, while in serum, especially MOC and GFAP were associated with outcomes reflecting disease severity, so reflecting different domains and different parts of disease activity and severity. And we think this is quite promising for the future. Thank you very much.
Wonderful. Thank you. This was 30 seconds. So, Sarah.
Okay, back to preclinical research, actually. So I'm here to show you how hematopoietic stem cells, so the cells that give rise to the whole immune system, can be influencing neuroinflammation. And basically, what we've shown using bone marrow, heterochronic bone marrow chimeras, is that by rejuvenating the hematopoietic immune system, meaning by transferring hematopoietic stem cells from young into aged mice, we can reduce the clinical score of the preclinical model experimental autoimmune encephalomyelitis. On the other hand, when we transferred hematopoietic stem cells from aged mice into young mice, we could increase the clinical score. And at the basis of these things, so what we think is happening is that the genetic and epigenetic adaptations of these hematopoietic stem cells that happen in aged mice and patients can be influencing monocytes and monocyte-derived cells that enter into the brain, migrate to the lesions, and can...
Yes. Thank you. And we come again to Alexandra Rodriguez.
Okay, thank you. So we've been discussing how progression in MS seems to be driven by CNS compartmentalized smoldering inflammation and how disease-modifying therapies currently on the market don't fully address it. This seems to be the case because therapies are focusing on adaptive immunity, whereas the smoldering inflammation is rather driven by innate immunity. So in order to address this research gap, our lab has been looking at innate pathway signaling, and in this case here I'm discussing toll-like receptors. So what we've done is that we knocked out toll-like receptors and their downstream adaptive molecule in constitutional mice, so constitutional knockouts, and then we induced them with a progressive mouse model of MS, so EAE. And then we did at various time points flow cytometry and single-cell analysis. In brief, our conclusions are that the TLR4 mediated TRAM axis seems to modulate the pro-inflammatory phenotype of myeloid cell, which in turns are going to lead to T-cell priming. So this by investigating these pathways, you're showing that innate immunity plays a definite role in smoldering inflammation and can be a potential target for the future. Thank you.
Thank you. And Sophia.
So it's just to go here, yes?
The green button, yes.
Okay, using Swedish MS code, we selected two extreme progressive MS cohorts based on their EDSS trajectories. And in both cohorts, progressors were matched 1-to-1 to non-progressors, and the progressors accumulated substantial disability during follow-up, while non-progressors did not. And by using the already existing serum biomarker measurements in SMSC, we saw that progressors had a significantly higher serum GFAP than non-progressors. And we believe that these cohorts are a prototypic data set to explore new biomarkers for progressive MS. And it's an ongoing project with Team mass spectrometry ongoing and further proteomic analysis planned. Cool, thanks.
Thank you very much. Okay, then we come to Sabine Schädele.
Hi, in this project we reconstructed TFP trajectories in all patients in the SMSC where we had at least 8 serum samples within the first 15 years, and we used an unsupervised clustering algorithm to cluster those. And if we associated these clusters with clinical outcomes, we saw that patients with an increasing or high GFAP trajectory, the patient who had increasing GFAP over time were at higher risk for PIRA events and also showed more pronounced cortical and spinal cord atrophy. In addition, they had higher MSSS at last visit. These results show that GFAP and also the GFAP dynamics over time are a promising biomarker, liquid biomarker for disease progression in MS.
Wonderful, thank you. All right, and then last but not least to Isa Slotboom.
So most patients with MS present with balance problems, and that's why balance training is a big aspect of rehabilitation. But the question remains, how do we train balance? Because training principles like intensity and progression are well established in strength and endurance training. However, it remains unclear how to apply these principles in balance training. So to address this gap, we designed a intervention that trains balance training individual to the balance capacity, monitors intensity, and systematically progresses the exercises to train patients to their limits. So if you're curious, please come visit me at poster 23.
Wonderful, thank you. So I think all of our flash speakers deserve once more a very loud and enthusiastic applause. Thank you very much.